السلام عليكم جماعة شحالي واعيين باشن كان الموضوع نيدا سبيكن أوجك أمينو أسيد and proteins موضوع جلا خوشة جالك مفايا وجالك بيزاني تدانن جالك بيزاني مهم هذا مستقبل جد تودي محتاج بين أما برابط تركيز السر ستركتشري كن كا ستركتشر أمينو أسيد بروتينات شوانا تفاعلات شو لوانا چيا وارتشتنا background. Proteins are the most abundant biological macromolecules occurring in all cells and all parts of cells. Proteins are biomolecules that can that's found in all parts of the cells, in the cell membrane, in the nucleus, in the ribosomes, nearly everywhere proteins can be found, and they are the most abundant biological molecule. The percentage of proteins is higher than all other biomolecules like nucleic acid, carbohydrate, and lipid. About 60%, 70% of biomolecules are protein, so the percentage is so high that shows and this shows that they are so important. Proteins also occur in great variety, thousands of different kinds ranging in size from relatively small peptides to huge polymers with molecular weight. So because there are thousands of different kinds of proteins, so the length of the sequence is different from each other. Some peptides can be built with 10 amino acids, some with 20, some with thousands, some with millions even. So molecular weight differ from one amino acid, from one protein to another. So the size is different, and all this will make the function of both of them different from each other. All proteins, all of them, whether from the most ancient lines of bacteria, proteins that are found in bacteria, or from the most complex forms of life, cells of animals and humans, are built from the same 20 amino acids, the same set of 20 amino acids. So here we have 20 different blocks to build a protein, 20 of them. There are some other amino acids, but they are called derived amino acids. They are derived from these 20 amino acids. Because each of these amino acids has a side chain with distinctive chemical properties, this group of 20 precursor molecules may be regarded as the alphabet in which the language of protein structure is written. So they are like alphabet, Arabic language. So in Arabic we have about 27 alphabet, in English 26 or something like that. So in a protein we have 20 alphabet. They are the same as alphabet. When you change the position of amino acids, the meaning of the word will be changed. So the protein will be changed, so the function will be changed. You see what I mean? فالفيري وكي حرفان خواندنا حرفا مثلا انت بيست هنا بيست حرف تهنا وانا هكا تجي حرفا بدل كده معناه بدل بد دي شو البدل بدل الفيري جي نفسي شو اليا مثلا تا ببتايدا كا بيكاتيش بيست امينو اسيدا ايك دي بيستا وموقع هردو كوكي ايك بس ايك جاواز بد شو الهمي دي بدل بد. From these building blocks, different organisms can make such widely diverse products: enzymes, hormones, antibodies, transporters, muscle fibers, the length of protein of the eye, feathers, spider webs, rhinoceros horn, milk protein, antibiotic, mushroom poisons, and all these are proteins in nature. All these are built from building blocks, which is amino acids. You see how important how important proteins are. Amino acids. So all these till now, all this was a little bit of background about protein. So now we will start with amino acids. Proteins are polymers of amino acids, with each amino acid residue joined to its neighbor by a specific type of covalent bond called peptide bond. If you remember, in polysaccharide, joining two monosaccharide with each other, the bond was called glycosidic bond. Here the bond to join join two amino acids together, the bond is called peptide bond. We have as we said earlier, 20 different amino acids are commonly found in protein. All the amino acids have trivial or common names, in some cases derived from the source from which they were first isolated. So we have common names and we have formal name, which is named in the literature. Asparagine was first, so we have amino acid called asparagine. They are called like this because they are first isolated in asparagus and glutamate in wheat gluten. Tyrosine was first isolated from cheese, and glycine was so named because of its sweet taste. Amino acids share common structural feature. So all these 20 amino acids share some stuff, and they differ from each other with some aspects. We will explain all of them in detail. All 20 of the common amino acids are alpha amino acid. All of them without exception are alpha amino acid, and we will explain what's alpha. They have a carboxyl group. All of them have carboxyl group, and an amino group. All of them have amino group bound to the what? Bound to the same carbon. Look here, all amino acid, all the 20 amino acid have structure like this. They have this hydrogen, they have carboxyl group, negatively charged, and they have amino group. All of them have this, and they have R group. They differ in what? They differ in this R group. All of them. If you change this R group, the amino acid will be changed. Here, this is alpha carbon. Alpha carbon. You see, this is carboxyl group, this is carbonyl group, and this is alpha carbon, and this is alpha hydrogen. Okay, we will name it alpha hydrogen. This, you see this structure, it's tetrahedral. All the 4 groups are attached to the same carbon. All the 4 groups are different from each other. They differ from each other in their side chains, or R groups, which vary in structure, size, and electric charge. So this R group may be changed from one amino acid to the other with the structure, size, electric charge. Changing each one of these will change the chemical nature of the amino acid, and which influence the solubility of the amino acid in water. For all the common amino acid except glycine, all the amino acid except glycine, the alpha carbon is bound to 4 different groups. I forgot to explain this. A carboxyl group, an amino group, an R group, and a hydrogen atom. And the glycine, the R group is, so where is it? All the amino acids, the alpha carbon, this is alpha carbon, are attached to 4 different groups. All of them, you see here, 4 different groups. 4 different groups. Isoleucine, 4 different groups. Leucine, 4 different groups. Except glycine, we don't have 4 different groups. You see, we have 2 hydrogen atom attached to the alpha carbon. The alpha carbon atom is thus a chiral center. Because of the tetrahedral arrangement of the bonding orbitals around the carbon atom, the 4 different groups can occupy 2 unique spatial arrangements, and thus amino acids have 2 possible stereoisomers. Since they are non-superimposable mirror image of each other, the 2 forms present a class of stereoisomers called enantiomers. All molecules with a chiral center are also optically active. So you see the alpha carbon basically is chiral center because it's attached to 4 different groups. They are tetrahedral, and this can be D or L. If you remember, in monosaccharides, all the monosaccharides were D. Did you remember? Here we have opposite amino acid. All the amino acid in the blood, in the animal, are L amino acids, and they are all optically active. The amino acid residue in proteins are L stereoisomers, step L. Nearly all biological compounds with a chiral center occur naturally in only 1 stereoisomers, either D or L. The amino acid residues protein molecules are exclusively L stereoisomers. In laboratory, we cannot synthesize only D or L. If we synthesize, for example, we synthesize monosaccharides or synthesize amino acids, we will have a mixture of D and L, and this mixture is called racemic mixture. 50% L, 50% D. But when chiral compounds are formed by ordinary chemical reactions, is racemic mixture, as we said, mixture of D and L isomers, which are difficult for a chemist to distinguish and separate. It's quite difficult to separate racemic chemicals. We have some chemical reactions to do that, but to a living cell system or a living cell, D and L isomers are as different as the right hand and left hand. Cells, in contrast to the chemical reaction in the laboratory, are able to specifically synthesize the L isomers of the amino acid because the active site. Why cells are capable to do that? Because the enzymes active sites are asymmetric, causing the reactions they catalyze to be stereospecific. Amino acids can be classified by R group. So we have classified amino acid based on the R group, based on the chemical nature of the R group. The topic can be simplified by grouping the amino acid into 5 main classes. Based on what? Based on the properties of their R groups, in particular their polarity. So we have classified the amino acids into 5 groups based on the polarity of the R groups. The first group is nonpolar aliphatic R groups. The R groups in this class of amino acids are nonpolar and hydrophobic. The side chains, like what alanine, valine, leucine, and leucine, tend to cluster together within the proteins, stabilizing protein structure by means of hydrophobic interaction. Glycine has the simplest structure, although it's formally nonpolar. Its very small side chains makes no real contribution to hydrophobic interaction. Methionine, one of the 2 sulfur-containing amino acids, has a non-polar thio-ether group in its side chain, Proline has an aliphatic side chain with a distinctive cyclic structure. This is non-polar aliphatic R group: glycine, alanine, proline, valine, methionine, isoleucine, leucine. So here, this R group, all of them are non-polar, all of them, and they are aliphatic. Aromatic R groups like what? Like phenylalanine, tyrosine, and tryptophan, with their aromatic side chains are relatively non-polar. They're also non-polar, but the R group here is aromatic. We have aromatic ring, benzene ring. All can participate in hydrophobic interactions. So here we had hydrophobic interactions. Here we also have hydrophobic interaction. The hydroxyl group of tyrosine can form hydrogen bond, and it is an important functional group in some enzymes. Tyrosine and tryptophan are significantly more polar than phenylalanine because of the tyrosine hydroxyl group and the nitrogen of the tryptophan in dol ring. Tryptophan and tyrosine are significantly more polar than phenylalanine. Why? Because of the tyrosine hydroxyl group and the nitrogen of the tryptophan indole group. In tryptophan we have indole ring. In tyrosine we have hydroxyl group. That's why they are more polar. Here, phenylalanine. This is the side chains of the phenylalanine. This is the tyrosine. This is the tryptophan. This is the indole group. You see, we have NH. That's what, and here we have OH. That's why this and this are more polar than phenylalanine. Polar. The third class is polar uncharged R groups. So we don't have positively charged or negatively charged. The R groups of these amino acids are more soluble in water or more hydrophilic than those of the non-polar amino acid because they contain functional groups that form hydrogen bond with water. Like what? Like serine, threonine, cysteine, asparagine, and glutamine. So here they are uncharged, they are polar, they are more polar than the previous classes. The polarity of serine, why they are polar? Because it's actually they have a functional group like OH, sulfur hydride group, etc. We'll see it in a minute. The polarity of serine and threonine is contributed by their hydroxyl group, that of cysteine by its sulfur hydride group, and that of asparagine and glutamate by their amide groups. These groups make this class polar. Here you see serine, we have hydroxyl group, hydroxyl here, sulfur hydride group here, amide linkage here. This is what make this amino acid polar. Asparagine and glutamine are the amides. So we have two amino acids that have amide linkage, like what? Like asparagine and glutamine. Only these two amino acids have amide linkage. Cysteine is readily oxidized to form a covalently linked dimeric amino acid called cysteine, in which the cysteine molecules are joined by a disulfide bond. Here we have, this is the amino acid cysteine. If these two molecules of this amino acid react with each other, oxidize it, cysteine will be formed. Cysteine. This is a dimeric molecule with a disulfide bond attached to both of them. The disulfide residues are strongly hydrophobic. So here the molecule was polar. Here it has become non-polar. You see, this disulfide bond make the molecule non-polar. Disulfide bonds play a special role in the structures of many proteins by forming covalent links between parts of a protein molecule between two different peptide chains. Positively charged R groups. So we have some amino acids that are positively charged, like what? Like lysine, arginine, histidine. The amino acids in which the R groups have significant positive charge at P7 are lysine, which has a second primary amino group at a position on a disaliphatic chain, arginine, which has a positively charged guanidino group, and histidine, which has an imidazole group. All these three amino acids are positively charged. Histidine is the only common amino acid having an ionizable side chain with a pKa near neutrality in many enzyme catalyzed reaction. A histidine residue facilitates the reaction by serving as a proton donor or acceptor. This is the structure of the three positively charged amino acid. Here, look here, lysine. Let's start with lysine. This is the R group. You see here we have NH2 that can accept an electron and proton and become positively charged. Here the same thing, and here the same thing. No, here can accept another proton and become positive. You see, this is positively charged amino acids. Negatively charged amino acid, like what? Like aspartate and glutamate. When we say positively or charged or negatively charged, we are talking in a medium with a pH 7. So of course, if you make the medium acidic, it will become positive. Or if you make the medium basic, it will become all the amino acids will become negatively charged. But here we are talking at pH 7. All of the amino acids are neutral except negatively charged amino groups, amino acids, and positively charged amino acids. Negatively charged like aspartate and glutamate, you see, the R groups have carboxylic acid group that can become negatively charged, and common amino acids or derived amino acids. They are also important amino acid. They carry out important biological roles. In addition to the 20 common amino acids, protein may contain residues created by modification of common residue already incorporated into polypeptide. So they are derived from the amino acid that we talked about. Among these uncommon amino acids are 4-hydroxyproline. So they are derived from the amino acid proline, a derivative of proline, and 5-hydroxylysine derived from lysine, amino acid lysine. The former is found in plant cell wall proteins, and both are found in collagen, a fibrous protein of connective. Collagen is very important protein in the bone, in the skin. Collagen is rich with amino acids, 4-hydroxyproline and 4-hydroxylysine. Selenocysteine, another derived amino acid, is a special case. This rare amino acid is introduced during protein synthesis rather than created through a post-synthetic modification. It contains selenium, the element selenium, rather than the sulfur of cysteine, actually derived from selenium. Cysteine is a constituent of just a few known proteins. Amino acids can act as acids and base. So amino acids, like water, they are amphoteric. They can act as acid or base. When an amino acid is dissolved in water, it exists in solution as the dipolar ion or zwitter ion. So remember this, in any amino acid, when we have negative charge, positive charge at the same time, uh, this ion, this molecule is called zwitter ion. They can accept proton and they can give proton easily. Okay, here it gives a proton. You see here, it gave a proton. Removing proton from here, and this is the product. Or it can accept the proton, as in this reaction, it becomes like this. This is a graph, a titration graph, titration curve of the amino acid glycine. Here, at pH 7, 0 OH, when there's no any OH, the structure of glycine is like this. See, because it's acidic medium. No, sorry, it's not pH 7. It's less than pH 7. Except proton, and the molecule is positively charged. When here, when it react with OH, hydroxyl group, become here, the OH will remove the hydrogen atom, this hydrogen, from the molecule, and the structure will become like this. This is called zwitter ion, and this reaction is pK1. pK1 is in this position. When we add further hydroxyl group, the structure will be like this. Okay, another proton will be removed from this amino group, and this is the structure. Here is zwitter ion. Here in the basic medium become negatively charged. In the acidic medium, all of the amino acids are positively charged. Titration curve predict what the electric charge of the amino acids. So from the titration curve we can predict the structure. We can predict the pK1, pK2, and we can predict the electric charge also. What's the charge of the amino acid? Is it positively charged, negatively charged, zwitter ion, or what? An important piece of information derived from the titration curve of an amino acid is the relationship between its net electric charge and the pH of the solution. At pH 5.97, the point of inflection between the two stages of the titration curve, glycine is present predominantly as its dipolar form, fully ionized, but with no net electric charge. The characteristic pH at which the net electric charge is 0. So when we have one positive, one negative, two positive, two negative. So here the net, be careful here, the net electric charge. When we have a 3 positive, 3 negative, 1 positive, 1 negative, the net electric charge is 0. And this point is called isoelectric point or isoelectric pH, designed by PI. Glycine has a negative charge at any pH above its PI and will thus move toward the positively charged, the anode. So when above PI, if you make glycine negative above pH, basic pH will become negatively charged. So in this case you are making glycine to move towards the positive charge, because it's negatively charged. It will move toward the positive charge, which is the anode. when placed in an electric field. At any pH below its pI, glycine has a net positive charge and will move toward the few less than pH that found in deuterion. If you make it acidic, less pH less than pI, it will become positive and it will move toward the negative electrode, which is called the cathode. The further the pH of glycine solution is from it is eligible with the greater the net electric charge of the population of glycine molecule. Amino acids with an ionizable R group have more complex titration curve with a 3-stage corresponding to the 3 possible ionization steps. Thus, they have 3 pK values. The additional stage for the titration of the ionizable R groups mesh to some extent with the other two. The titration curves for 2 amino acids of this type, glutamate and histidine. So here it's a little bit more complex. In glycine we have 2 pK, pK1, pK2. Here in for glutamate, and this is the structure of glutamate, positively charged, it can accept, it can gives a proton from here at the beginning, become dead like this, and then gives another proton from here and then another proton from here, you see, and become a 4 pK. Here in histidine also we have 3 pK values depending on the structure, and this is pK1, pK2, pK3. We have 3 S shapes, 2, 3. Peptide and protein, we will leave this to next lecture.